Hey girls, how are you doing? So happy Friday. Only a few more weeks of virtual school left. I just wanted to pop on, let you see my face. But I just wanted to pop on and kind of explain a little bit about mitosis and the cell cycle, and kind of just clarify a couple of things that I've been seeing based on what you have said in your fairy tale drawings. So first of all, Khaleesi, please get out of our way. So first of all, what you're currently studying is the cell cycle plus mitosis. So the cell cycle describes what is going on inside of cells most of the time. So you know that the cell cycle consists of interphase, which includes G1, which is growth 1, S, which is synthesis, and G2, which is growth 2. And then after interphase ends, if the cell is able to pass certain test points, essentially, then the cell will enter into mitosis. Mitosis occurs in somatic cells, which are body cells, so it does not occur in your gametes. So for all of us, we're all females, so we all make egg cells. That's our gamete. And in males, they have sperm, that's their gamete. Okay, sperm and egg cells do not undergo mitosis. They undergo a process called meiosis, which we will talk about in our next little unit. So interphase comes first with G1, S, and G2, and then you have mitosis, which consists of PMAT, which is prophase, metaphase, anaphase, and telophase. Now, some scientists will argue on whether cytokinesis, which is the full splitting and forming two new cells, scientists will argue on if that's part of mitosis or if it's just part of the general cell cycle. For our class, we'll say that it is part of mitosis because you end up having two completely new cells by the end of cytokinesis. Cytoplasm. Kinesis means movement, so you're splitting the cytoplasm and the cells are able to move apart. Now, I'm going to flip to share my face and also my screen. And I'm going to show you guys what is going on in each step of mitosis. And I'm actually going to show you all 46 of the chromosomes because sometimes we think we understand what we're doing when we see just like one or two chromosomes, but really it helps to see all 46. So I drew some pictures and I'm going to share those with you guys, and I'm also going to put it on the screen so you can see that too. I know the language used for chromosomes and chromatids gets a little fuzzy, which is why you had that whole section on the WebQuest about what all those different terms mean. Basically, you have three main types that we'll talk about. And these are all just forms that the DNA is found in. You guys already know what DNA is made of. You know that it's a deoxyribose sugar. You know that it's a nitrogenous base. And you know there's that phosphate group. So that sugar phosphate backbone and then the nitrogenous base. So you're going to have just all of your nucleotides making up your DNA. And then we give the DNA different names depending on what part of the cell cycle we're in. So when the cell is in interphase, it's basically in the form of what we call chromatin, which is basically just like long strings of spaghetti. It's really messy. It's not very organized. It really is kind of just a mess. That's chromatin, which is still just made up of DNA. And then you have during prophase, really the end of G2, beginning of prophase, you have your chromosomes start to condense. So all of that loose, stringy chromatin condenses down into what we call a chromosome. So it just forms the chromosomes, which is what you typically think of when you think of like those little pictures of like little X's that you've probably seen in like Grey's Anatomy and other scientific shows. That's a chromosome. It's just a condensed form of the DNA. The reason why we have to condense the DNA is because we're about to start separating it into two new cells. if it was just in loose chromatin form, it would be really difficult to make sure that each new cell ends up getting all of the DNA that it needs. So we condense it into the chromosome form to make it easier to separate. That is literally the only reason why that happens, is so that it's easier to separate and keep track and make sure that each cell has the exact same DNA. Because remember, mitosis is occurring in your somatic cells. That's what's making more of you. More of your skin cells, more of your lung cells, more of your liver cells. Not all somatic cells are going to undergo mitosis, but most of them do. An example of some of them that don't is things like your nerve cells. So when you think of people becoming paralyzed, that's because once those cells are damaged or killed, they don't make more copies of themselves. So once they're destroyed, which is why you can recover from a cut on your arm, but you can't regain the ability to walk after you're completely paralyzed. It's because those cells do not undergo mitosis. Some other cells that don't go through mitosis would be like your red blood cells. Red blood cells actually go through a process called hematopoiesis, which takes place in the bone marrow. And that's not mitosis. It's a different process. We're not going to cover it, but you can look it up if you want. And another type of cell that does not undergo mitosis would be your muscle cells. I think we've talked about this in class before, but your muscle cells actually, they don't multiply in number. The reason why your muscles get bigger is because the cells themselves will lengthen and get bigger, and you'll have all of these different fibers that help to make the cells bigger and help make your muscles bigger. So that's the actual reason why your muscles get bigger. It doesn't have to do with more muscle cells. So like you and like, maybe not Arnold Schwarzenegger, he probably does have more muscle cells than you, but you have relatively the same number of muscle cells. People just work out and make their muscle cells larger. So that's kind of cool. But the whole idea of mitosis, again, is to make more of you. So things like repairing cuts, repairing internal organ damage, any type of repairing, that's mitosis. Making more of you so that you can grow and go from being the size of a baby who's like 6 pounds, 12 ounces when they're born, to being like an adult that's between 100 and 350 pounds on average. So you have to make more cells to do that. But the main thing that's going to separate mitosis from the next process we talk about, meiosis, the main thing that separates those is the point of mitosis is to start with one cell and end up with 2 identical daughter cells. They will be exactly the same. If they're not exactly the same, something went wrong. Either some of the checkpoints didn't get passed correctly. Cancer cells have a really great ability to overcome those checkpoints. And so that's why those cells become harmful, because they just can make copies of themselves over and over and over again, even if there's stuff that's going wrong. And so those cells end up not being able to function properly, which causes the symptoms that you get when you have different types of cancer. But the overall goal, if everything went the way it was supposed to, the goal of mitosis is to end up with 2 daughter cells that are exactly the same as the parent cell was. So I think that's kind of all the ranting and rambling I have to say before I show you guys these pictures. So I'm just going to pause the recording real quick, so if there's like a weird a glitch or I look like I'm in a different place whenever I resume recording, I'm going to pause real quick so I can pull up those pictures, and then I'll start recording again. Okay, so joke's on me. This software does not allow me to switch from one type of recording to the other. So I'm going to stop this video here, and then I'll upload right behind it the one that has the pictures. So my bad. But what I say in this video is still important, and you need to know it. So